reach that platform in a very short amount of time because they don't like to swim, so they like to be rescued from there. And they do that in a very spatially precise way, meaning that even if we take out the platform on the very last day, they're able to persistently look for the platform where the platform is supposed to be, which tells us that they have really made an association between the specific place of the environment where the platform was and the ability to escape. And they use this spatial layout that is encoded into the hippocampus and the internal cortex to reach the specific location. This is something that Maria has already explained. I'm not going to go further. And you have also seen some example of this through your videos. So you should be well versed in that. But solving this task means answering a question which is not really trivial. So how can the brain know where the animal is? So how can it create and use a cognitive map by only looking at the activity of plays in grid cells? So we have seen before, and this is, if I remind you, this is what we have discussed before, that a certain stimulus induces a certain response in neurons. This is the encoding problems, but the brain needs to use the response of this neuron in order to try to understand what happens and what stimulus is driven that. This is the decoding problem. Try to extract information about the stimulus, about the neural response. So there is a problem with place cells, right? If the brain was trying to read the signal of the place cells or of the grid cells for what matters, and try to infer where in the environment the animal is just by the activity of the place cells, it would be very difficult because it could say precisely where it was only when the animal was in the location where the place cells fires. Imagine, for example, that the animal is running onto this treadmill that can create a linear map of the treadmill that is symbolized here. If the animal is in this location where the place cells is fire, it can comfortably say the place cells is fire, therefore I'm in this part of the linear track. But if it was in a play part in which the animal, the place cells was not firing, then of course the animal could be in this part of the track, but could also be in this part of the track. So it becomes extremely complicated for a brain or for an outside reader that reads only from the activity of one place cells that the animal is in a specific location just by one place cells. At the same time, you can see something that is very true for higher cognitive area, meaning that these areas are modulated, as I said, not from one individual sensory stimulus, but from the individual sensory stimulus and other variables that are correlated to the internal state of the animal, like arousal, attention, the emotional state, and so on and so forth. So if you look at every time the mouse passes through the specific part of the environment where the place cells fires, you see that the response of the place cells is quite variable. The first time that it passes, it fires a little bit. The second time's more. The third time's less. Fourth, fifth, fifth more. But then there are two trials in which the animal passes through that specific location, and the place cells is almost silent. Sometimes it's very, very active. Sometimes it's quite almost silent. Which means that if you, on an instant-to-instant basis, were relying for your life to know where you are only from the location of one individual place cells, you really would be screwed because you have intrinsic variability, you have the possibility to sample just a small region of the environment. It's not really efficient. So the solution that the brain came up with and that we are starting to explore in modern neuroscience during these years is to listen to many of them at the same time. So that because you have place cells that are active in each and individual position of the environment, if you were able to listen to more than 150 place cells here, you would be able to identify in which location of the environment you were, not by the firing rate of one individual neurons, by the identity of the neurons that were active at that time. For example, if you were to be listening to cells number 1 and to 10, for example, to be active, you knew with a certain precision that you were in this part of the track. If you were to listen to this group of neurons instead, and these would be the ones that are active, you knew that you were in the middle of the track. And the same things here. If these cells out of the 150 were the ones that were active, you knew that you were in this position of the track. So even if individual cells sample a small part of the environment, you would be able, or if they have an unreliable response or a broad tuning curve, you would be able, by combining the message of many of them, to actually increase the precision of your decoding, of the decoding of your message, and then be confidently able to tell what stimulus has been driven your neural representation, which is what stimulus was actually presented to the animal when the network or the group of place cells was active in that particular combination. So these represent a sort of a paradigm shift in the way in which we study the brain. If you remember this morning, I told you that when we wanted to look for the neural correlate of Jennifer Aniston, we would look at individual neurons that are able to modulate their activity when the image was shown. In this case, we are looking at the population of neurons, and we are trying to decode the information about the stimulus, but the combined activity of neuron number 1, 2, and 3, and 4 in this case. We don't listen to just one cell, we listen to many of them. And this is extremely important because you can imagine that the pattern of activity that you have here is multiply in an exponential fashion, because you can have situation in which you can represent different information according to different gradation of activity of each one of these individual neurons. Okay, so we go from one cell to the population. We're starting to think that the neural code and the tuning curve of individual neuron might be less important for a brain that is able to listen to thousands of other cells, as we have seen on Monday, than what the combination of the inputs of the different neurons is actually signaling. So why and when is a population code advantageous? Well, it is advantageous when individual neurons are able to sample only a small portion of the stimulus space. And we see this with place cells. A place cell is active in a small part of the linear track. So you can say where you are by the listening to only one place cells, only if you're in that specific track. And at the same time, as we have seen today from the visual stimuli and the visual cortex, individual neuron might have broad turning curves. So even if the neuron is highly active here, the activity that he has for bars that are oriented at minus 15 degrees or at plus 15 degrees is still quite high. And being able to actually distinguish these two different orientation might actually be difficult. So by listening to many of them at the same time, you can actually understand better what the message is, and you can increase the precision of your conclusion, of your decoding in a very significant way. Okay, we move from individual neuron to population of neuron. And this is the basis of decoding that both our brain does and also our computational algorithm do. If you were in an invertebrate, for example, like Drosophila, in which you have very few neurons that encode specific message, sometimes up to 1 or 2 neurons, then the population coding would really not work, because you have only that neuron that is able to transmit that specific part of information. Having gone higher in the evolutionary timeline, we think might have been advantageous and having enabled organisms with higher cognitive function, because you can encode multiple states with a larger fraction of neurons. And the state would be encoded in the activity of the individual neurons taken as a population ensemble. Any doubt or question about this part? Because now we're going to see how this is advantageous actually in a real case of place cells activity, and how this can bring us to understand phenomena that we could not possibly identify by individual neurons. Okay, so this is a recording that has been done of a rat moving onto a linear track. You see that the linear track here is quite convoluted to be quite long, so that you can record many grid cells. And in the many place cells, sorry. And in the previous movie of a place cells that I showed you, you were listening from only one place cells while the animal was moving around. In this case, you will be listening to many place cells, and you see here the individual clusters in our tetrads recording for the individual place cells. You will see here, every time one of these cells is active, one spike will be overlaid in this ongoing activity plot. And here you will look at the spatial profile of the spikes while the animal, which is symbolized by this green circle, is moving along the linear track. So this is what a real cognitive map in the hippocampus looks like. Sorry, and of course this is not working right now. Okay. So we're following the movement of the animal. The animal is moving and sometimes is resting just to pick up some cookie crumbs and eat. And there is something that you will see if you look at the ongoing activity. So sometimes you have just one neuron that is active here, for example, now the red, then a little bit the blue, now it's the blue prevalent and so on. But there are moments along the track in which all the clusters and all the cells light up at once. And you see that this cluster of a yellow neuron was not present until the animal reached this last turn. That is the specific position of the environment where a place cell was active. So in the end, if we were to look at which individual neuron were active, we would see that there is this blue neuron here that is active in this position of the environment. There is this other light blue here that is active at this other position of the environment. The red is quite active here, but it has some also fighting in some positions. The yellow is active here, the green is active here, and so on and so forth. Of course, you also see that there are a lot of times in which the neuron is active, but not in the specific location where you would define their place field. For example, these blue spikes here, there is some spikes that is also outside of that. Or you have some yellow here, but you also can see some yellow, for example, in these clusters here. And there were some moments, as I showed you before, in which all the cells were active at the same time. For a long time, they thought, okay, these are mistakes. The place cells sometimes think that they are in a different position, but they're not really doing something that is meaningful. And we should just focus on the behavior of the cells that are basically in their place field. But this is not the case. And I'm going to show you later how this has been actually, how this phenomenon has been actually identified and actually considered as an important phenomenon for the way in which we use the cognitive map to find a way in the environment. The first thing, though, that I would like to highlight from this type of plot is something that is like here, right? We can model and say, for example, that every time the rats move, as it was in my previous animation, you find that the red cell is active here, the pink here, the yellow here, the light yellow here, and so on. But when you go and look at the real pattern of activity of the individual place cells, you see that the firing is much more spread. Here we define the place field of this cell in this specific location of the trajectory, but you can see that the cell was starting to fire here and continues to fire here. So on a cell-to-cell basis, the precision of each place cells is actually quite small. You wouldn't be able to decode that the animal is in this position only by listening to the green cell. So, of course, John O'Keefe has encoded has resolved the encoding problem by telling you that indeed the firing of this neuron is modulated by the position where the animal is in place. And that's how he found the grid cells, the place cells to begin with. However, since individual neurons represent information from a small portion of the environment, as I told you before, and also they're quite noisy in a sense, they start firing before a specific location and after a specific location. How can you go from listening to this particular activity to understand where the animal is in place? Take this example. If you read the green cell from this particular window of time, or in this particular window of time, you will find in both cases two spikes. Here, they're very quite close to each other. Here, they're quite distant to each other, but still quite close. When would you say that the animal is, if the only information that you had available was actually these two? It could be either here, or it could be either here, in two different parts of the trajectory. It wouldn't really be very, very precise, right? So listening to one place cells might not lead to an estimate of position. In this case, though, if you enlarge the window of neurons from we read from, that is, we listen to all the place cells, we will see that in this portion of the environment, the green cell is active, but is active together with the yellow, the pink, and the red cell. And in this portion of the environment, the green cell is active just as much as it was active in this portion of the environment. But this time it is active with the yellow, with the blue, light blue, with the blue, with the purple, and the pink one. So you see that by reading at multiple cells at the same time, and you don't need many of them, you actually can do precise decoding from 15 place cells. You can actually be able to distinguish pattern of activity that are associated with an earlier phase of the linear track or with a later phase of the linear track. So in the end, you can decode the position of the animal by simply understanding which patterns of activity are associated to a earlier part of the track or to a later part of the track by listening to multiple neurons at the time. This is usually what we do with computational method. We take the individual snapshot of activity of the population of neurons, not of individual neurons, and we tell to a decoder, usually is an algorithm, is a classifier, but we will see on Friday how we can use deep neural network for that this pattern of activity belongs to this part of the track, and this pattern of activity belongs to this part of the track. And once we have basically trained the decoder to do that, the decoder will be able to tell us exactly with a very high precision when the animal is, just by listening to a population of 10, 15 place cells. So what we do is usually like this. We record the cells and calculate their praise field, as we have done before. We listen to one individual neuron. This is the encoding step of decoding place cells position. We'll split our sessions in half. One half of the session is used to tell to the encoder, okay, when this pattern of activity is present, then the animal is in this position. And when this pattern of activity is present, then the animal is in this position. And the encoder will start to make the connection that when these cells here are active, the animal is in this position. But when these cells here are active, the animal are in this position. At this point, we test our model, and the model use the second part of our recordings to test that what actually is learned is correct, and it can decode the position of the animal only looking at the neural activity of the animal. Then we validate our model on a series of data that are not labeled, so new data that are untested that we use to test our hypothesis and our algorithm. And we use the classifier that we come with on new recordings. And we can reach a level of precision that is staggeringly high. It's higher than actually the precision of one body length of the mouse. For a mouse, it arrives to a precision of 5 centimeters. What I mean is in this case, you see here a certain number of place cells that were fed to the decoder. This is 32 place cells that were recorded at the same time. You see here in blue, the line in blue is the actual position of the animal along the linear track. And in red is the position the decoder trained on these place cells is able to predict at every instant. But just looking at the activity of this neuron. You see, of course, that there are moments in which the decoder is not extremely precise, but overall, the precision is quite high. And again, if you calculate how big the error is between the calculated with the predicted position by the decoder to the actual position of the animal, you find that this is really higher than the body length of a mouse. It's 5 centimeters, where the body length of a mouse is actually 10 centimeters. I would say that is a pretty good job. At the same time, you see how individual neurons would be quite noisy if you just were to listen to the type of spikes that they were actually emitting. Okay, so if we go back to the movie that we have seen before, we use the same recording to train a decoder based on Bayes' theorem that we're gonna see on Friday to estimate the position and the direction of the animal. You can estimate that the position is moving forward or the position is moving backward and the position where the animal is. So in this movie now, you will see the green spot is always the position of the rat. The arrows are the prediction of our classifier. When the arrow is gonna be blue, the predicted direction is forward from this end of the track to this end of the track. When the arrow is red, the predicted position is actually moving backward. So let's see if our decoder is actually good. So one thing that I really want you to pay attention in this movie is that you see that when the animal is moving, is actually quite good, right? When the animal is moving, the error of the predicted position is actually very close to the green spot of where the rat is. But when the animal is stopping or is looking on the side or just sampling the environment, then you see something quite strange happening. You see that these arrows of the predicted position is starting to jump all over the place. Most of the time, it's not really something that is completely random. You see that there are instances in which the arrows jump ahead, or instances in which the rat jumps, the arrow moves behind, but always following the path that the rat was making. This is what has been discovered through that position before. So by simply using the decoder to be able to decode the trajectory of the animal from the activity of the animal, we can ask one simple position, that is, what happens in the cognitive map while the animal is not running? Is the animal thinking about different position where it is supposed to be or where it might be? So is the cognitive map actually trying to predict where the animal is going to go, or remembering where the animal is just been? Which is basically like how you can remember the past or imagining where you're going to go into the future. And this is what I've seen before. If I now stop at one of these moments in which the rat stops, for example, here, you can see that the animal now is trying to go on the other part of the path, of the path. So, and this moment in which the position that is predicted jumps ahead or behind the animal are moments in which many of these neurons are active at the same time. Those are the spurious spike that I told you before. So the place cells in the end ends up is not only active when the animal is in one position to signal that the animal is moving to that particular position, but when the animal is resting or is scanning the path in front of itself to try to plan the road that is going to walk on in the next few seconds, in a phenomenon that is called replay or pre-play, in some of these cases, right? So the cognitive map actually samples position in the environment in which the animal is planning to go in moments in which the animal stops. And the animal plans the trajectory ahead, for example, toward a certain goal, by replay in a very fast fashion the trajectory of place cells that will lead him to reach that particular reward. And you can decode when this event happened by simply like looking at the activity of the population of neurons and seeing which neurons are active and with which firing rate through a decoding algorithm as the one that I just showed you. But this is not the end of the story. You can also do another experiment. You can put your rat into a sleeping bag or a flower pot in this case. You can continue recording spikes, and you can see that when you do that, the animal is going to be dreaming about walking through this path. The neural representation is starting to describe paths that move forward or behind along the path in a very coordinated fashion. And if you see, this is not completely random. Like, it's not that you see this period lighting up and then this moment lighting up and then this places combination lighting up. If I replay it constantly, you can see that this is actually following some trajectory that go either forward or backward in place. So there was a representation of the rat going in this direction in this case. And now there is another representation of the rat moving in through this direction along this part of the environment. This is a phenomenon that has been discovered through decoding algorithm that basically highlights how we are constantly replaying experience that we have done during our exploration when we are actually resting. And this is very important, and it's something that we will see tomorrow and that we are going to be seeing also on Friday, because it's a way in which we consolidate information, in which we revisit the experience that we done during our conscious experience to transform this experience-related activity into memories. Okay, so neural decoding is one of the most promising approach to try to understand the hidden messages of neurons. This is the sales pitch that I need to give you about neural decoding. Right now, it is used for a lot of different type of application. Some of these are trying to understand what type of information is encoded in the activity of population of neurons when we have not a very clear idea of what messages actually this neuron encode. And we might basically have no idea of what this message this neuron might encode because, for example, we have no anatomical or lesion study that guides the way at what type of information might be processed from this neuron. The variables that might be processed by a specific neuronal population might be actually hidden to us, for example, emotion. Up to very recently, we could not really gauge if the animal was expressing fear or like disgust or pain or surprise in many different ways. This is very difficult from what we do in humans, for example, that are able to express a conscious perception of emotions. or because the responses might not be beautifully tuned to one specific stimulus because of noise or selectivity to multiple variables. That is what we have seen before with the place cells, but also what we have seen today with the cells that were tuned to specific sensory and visual stimuli. Okay? One case that I would like to bring to your attention about where decoding can help us understand what information is actually encoded in the activity of neurons is the last area of the internal hippocampal circuit that we haven't really touched upon today. And this is the lateral internal cortex. For a long time, the lateral internal cortex was sort of a mystery because, the middle internal cortex codes for space, the hippocampus codes for space, the subiculum codes for space, or at least there are spatially modulated tuning curves. What the hell is the lateral internal cortex doing? If you put animals in two different rooms, the firing of individual neurons is not so different from one room to the other room. There is not that beautiful place modulation that the other areas of the circuits seem to encode. However, last year or 2 years ago, some researchers have done a very clever experiment. They have put the animal first in room black 1, then in room white 1, white 2, black 2, black 3. They have put them alternating in the black room and then in the white room, and then in the black room and then in the white room, just to understand what the LEC might decode for. And then we fed to that decoding algorithm different variables to try to understand what the activity of this neuron was actually tuned to. And it turns out that the LEC is lousy at distinguishing if the animal is in the black room or if the animal is in the white room. But what it can distinguish very, very well in the activity of the cells is the time at which that room and that specific experience happened. If you look at the activity in CA3 or in the activity in the MEC, in CA3, you're very good at distinguishing if you're in the black room or if you're in the white room or if you are in the black room. In the LEC, it's a continuum. But experiences that happen earlier during the sequences are clearly separated by experiences that happen late during experience. So if you train a classifier to try to decode a variable from the activity of the neurons in the lateral internal cortex, what you can get information from is actually the time at which the experience happened and not really the identity of experience. And this is extremely important from what we are going to be talking about tomorrow, about the way in which the hippocampus might actually get information about space from the middle internal cortex, time about the lateral internal cortex, and combine them together to create a memory of a specific event that has happened in a specific space at a specific time. And these also come the idea of time coding in the hippocampus that your colleague was asking before to slide. But there is another, and this is really the last two slides of today, there is another field in which decoding the activity of neurons might actually really be valuable. And this is mind control, meaning that if we were able to record from activity of neurons while we are thinking about something and understand what actually those neurons are thinking about, we could use that neural activity, for example, to drive and control machines. I know that this sounds like sci-fi, but this is actually something that is quite advanced in the field of system and computational neuroscience, and is used to control those that are known as brain-machine interfaces to control prosthesis, for example. It is quite let's say, a successful approach right now to implant some microelectrode in the area of cortex that is able to basically represent movement and motor signals, to record the activity from these areas of cortex, decode what actually pattern of activity is when a person is thinking of moving his arm into a certain direction, for example, forward or at 90 degrees or 180 degrees. Use computational strategy to transform the activity of those neurons to specific trajectory in space of a prosthetic arm in real time, and use this information to actually move prosthetics arms in people that otherwise would be tetraplegic. So this is something that is actually, not sci-fi. This is an approach that is quite successful in many companies, and there is a lot of experimentation to actually develop this BMI further. So much so that last year it was published a new type of device in which electrodes have been implanted in people that were unable to speak anymore, in order to induce basically the same type of logic to the production of speech. So a certain electrode array was placed on the area of cortex in the brain of patients that is basically like responsible for producing speech. The activity of this neuron had been recorded when the person was thinking about pronouncing a certain type of words, and that had been used to basically like induce the production of that specific spoken language. So you see how neural decoding is important because it can not only drives our understanding of the brain, but it can drive new way in which we can treat some sort of very debilitating disease, like for example, paralysis, or like for example, the inability to speak. All right, so we arrived to a few points to sum up what we learned today, which was quite an intense day from the point of view of concept and the type of neural coding that we are basically like now exposed to. We know that neurons can encode information about the stimulus in the temporal profile of their activity. In the sensory areas, neurons are tuned to feature of the word, and they show their tuning through a specific tuning curve, and get activated only when a specific portion of the sensory environment is actually stimulated. This is the receptive field. Away from sensory areas, features get combined, tuning curves become more and more abstract. In particular, in the entorhinal hippocampal network, there is tuning curves that are locked to the spatial location where the mouse or the rat is, and or the speed at which the animal is moving. And this is something that I didn't mention before, but of course, we study these things and we deem important because the similar correlates have also been observed in the human brain. So we don't think that this is only something that regards mice or rats. We can study the cognitive map by means of teaching an animal to perform a spatial task like the Morris water maze. Tuning curves in high-end cortices are created by local network topologies. We have explored this with the idea of the attractors, and we can decode message of the brain from the activity of population of noisy and imprecise neuron. And that drives our study of the brain, but also drives the development of engineering products that help us to move machine with just our thought. And for that, this is more about the brain and to control machines. And that closes what I wanted to show you today and the lecture of today. This is the day in which we start to talk about the neural code and the neural correlates of cognition. Tomorrow, we're going to go through another function of the hippocampus, which is the formation of memory of everyday experience. And we are trying to understand what method we can deploy to actually test the hypothesis that we are formulating by listening to the activity of neurons through electrophysiology or calcium imaging. Do you still have you with me? I didn't lose you. Can I decode your activity? Is there activity? Good. So there is no question in Slido. Do you guys have any question for me? Anything that you want to discuss or comments on during of things that we went through today or from yesterday? Yeah.
When you talked about the prosthetics, I realized one thing. Because when you perform a task, you have a specific pattern lights up, and this is read out by the sensor to then perform the motion of the prosthetic. But when you remember something or imagine something, the exact same pattern lights up. So is there a way to differentiate imagining a task and actually performing said task, or is it just intermingled?
This is a very good question, to which I have to say that I need to use speculation because I'm not an expert of that, and I don't know if there is a moment in which, for example, you need the prosthetic to be online to be able to actually, you have to consciously decide that you want to move the arm, so you have to switch that on. But one thing that I can tell you is that when you are imagining something, the pattern of activity that you see appearing in the brain is related to the one that appears when you're actually doing something is much faster. So the temporal scale is much faster than what you actually do when you're performing a certain action. For example, before where you saw the movie of the play cells, you saw that all those neurons were active at the same time in a very small window of time. So you could really like select if you're performing a task or if you're thinking about the task by the temporal dynamic, the temporal pattern of activity dynamic that you're looking at. But I have to say that this is actually a very good question, and we can think that there might be ways in which this can be implemented, but I'm not aware of stuff like that.
Okay, thank you.
Thanks. Any other question about this? Done for the day? Okay, so I'll see you again tomorrow morning, 9:30, for going through the second to last day of this block course. Good? See you tomorrow. Bye-bye.